speeches as well as prize acceptance speeches that are all, I think, very interesting to watch. And again, when the Nobel Prizes, of course, are always for really important things that change in a broad way our concept within those fields. And this is one of these ones that, now again, on the one hand, we've known for a long time that microorganisms cause disease, but what was very odd about this one was that they really had to prove what we typically think of as a disease because of a entrenched dogma about stress and drinking acids as the cause of ulcers. Now again, I didn't know this before until I was doing some literature searching for this talk, is that there was actually a Greek physician who was treating peptic ulcers with bacteria, with antibiotics. And so again, the link is here, the link is also in the note card, but again, this was actually in 1958. So again, there were people who, in a very pioneering sense, were able to understand and treat the disease, but of course, as far as to know, he didn't work to actually isolate and to show through standard medical norms what the specific isolated causative agent was. So and again, it would also not surprise me if other people were treating this with antibiotics, but this is one I just happened to come across in my searches looking at the history of this. Learn something new every day. So the story, though, I don't think is at its most interesting if we were to end at the Nobel Prize, because again, reading through the text here, and this is text coming from the Nobel Prize press page, so a lot's been done since then, that Helicobacter pylori is common in people's stomach, even though ulcers are not. And so about 50% of all humans have recognizable, isolatable infections of Helicobacter pylori, but really only 10 to 50% of those people come down with any sort of disease. So this is, in terms of breaking with, again, in a very old dogma, breaking Koch's postulates. Because again, Koch's postulates would say anytime you have this bacterium, it will cause disease. And so what this particular bacteria, I think more than really anything else, maybe other than the vitamin K and E. coli story, started bringing us to realizing that there are infectious pathological organisms, but that sometimes they don't always cause disease. And in fact, there's an even bigger and greater story with this, as has been slowly examined. So again, one thing now that people have been doing is, now that we know antibiotics kill Helicobacter pylori, and we know that a large percentage of people have Helicobacter pylori, can we, from an epidemiology standpoint, examine other aspects of health and look at the associations between those? And so one of the people that's one of the trailblazers in this area is Martin Blaser, and this is from a review that was 2006. So again, there's a history of work prior to this. And again, you might need to zoom in a little bit on the picture to see all of the specific points here. But that, in essence, people have shown that Helicobacter pylori, based on genetic sequence analysis, have been infecting humans for at least the past 100,000 years. And so we have, at least for that amount of time, been co-evolving. And in fact, there are Helicobacter pylori that are also resident in chimpanzees and other close related primates. And so, again, with that type of long association, there are ways that evolution will, again, select for different things. So Explorer asks an interesting question, is it heredity? And I think I'll presume that the heredity question has to do with, if you have an infection, what is your additional susceptibility to actually progressing to an ulcer? And I think that's a big part of it. Again, this was kind of the thing that, with Blue's talk last week, which was a wonderful introduction to the wide variety of things that affect us, is that, traditionally for a long time, microbiology departments also typically are called microbiology and immunology departments. And that's because there's this very intimate association with how we understand both. We have a very robust understanding of immunology, and that there are a lot of factors that make our immune systems different from each other. both in terms of the so-called innate immune system, which is something that's always on, always ready to attack, always ready to be on the defense. But because it's always on, if you have bacteria that do live on you all the time, you have to make sure it doesn't attack them. And then there's also the adaptive immune system. And the adaptive immune system, again, also is very good and gets trained to fight off infections, but you also have to teach it to not overreact to things that are always there. And in addition, there's this whole suppressor area of the immune system that helps regulate a lot of these. And so there are a lot of genes, a lot of players that are involved not only in developing immunity, but also suppressing our own immune system to things that aren't harming us. And it's horribly complicated, and maybe Blue or I or somebody will do a talk on immunology one of these days. And you know, and Abby asked, how does one teach the immune system? And I don't with microorganisms specifically, but basically the current understanding is that as we are born and as we are exposed to the outside environment, our body learns what's okay to have on us and what's not okay to have on us. And that largely has to do with the additional idea of, again, a microorganism on our body doesn't necessarily cause us any harm, but if it does something like starts disrupting cells or starts secreting toxins, then we react to it. And so on another sense, the bacteria have to learn to behave when they're living on us at the same time. So there's this coevolution and cohabitation that takes place with a lot of these. And yeah, there actually is an association of diabetes and obesity related to microorganisms, although I don't know specifically about type 1 diabetes, as Explorer's mentioning. So again, let me just summarize this slide here. And this is a summary of, again, Martin Blaser, some of his work, as well as work by others. Again, there was a talk, the New York Academy of Sciences also additionally is the basic publisher of this work, which is actually a nice release into the Creative Commons. And so, again, what this diagram is showing is that we've lived together for a long time, and that when we have Helicobacter pylori in us, then on the left side, you see the little circles with little positive signs of peptic ulceration, as well as gastric cancer. And what he's summarizing from a large amount of work, looking at epidemiology, looking at, again, infection rates of a variety of things around the world, is that our risk of getting other things actually decreases. So in terms of asthma, in terms of allergy, in terms of diarrhea, in terms of GERD, which is gastroesophageal reflux disease, which again is something that promotes esophageal cancer, as well as things having to do with energy imbalances. So again, having to do with obesity rates. And again, Simulat staging where this is going, and the thinking of this is that we really represent on our bodies an ecosystem more so than, again, a solid mass of animated supernatural matter that's infused with life. Okay, so now I'm going to get a little bit data heavy, so bear with me. I'll try and explain the slide as best as I can. And again, in this summary, this is an example of one individual infant's diversity of microorganisms, and I think it's primarily in intestines, but maybe it includes intestines and stomach. And what's represents time, as well as a few important events in that particular child's life. And then what's on the y-axis, the vertical axis on the left, is from lowest to highest, going from, again, the downside to the up, is the amount of biodiversity seen in its gut. And so you'll notice that over time, again, one of the major trends, of course, is that over time, from birth until a few years old, that it's increasing. And that is something that is established by interactions with a variety of sources. Look really close at the text. The child is primarily breastfed. And so again, the stuff that's in those ducts, the bacteria on the surface of the breast, those are things that are getting ingested. But it's also from the household. Again, there's lots of associations of the bacteria people have when you get a new roommate, for example. Or if if you're living with different cousins or different people coming in, your microbiome starts to resemble the people you live with and what you have in your house environment. Now there is one event here that's important to recognize, and that's what we have here on the next slide, is again the green arrows pointing out a time in which the child received amoxicillin. And I didn't go through and delve into the cause, the reasons for getting amoxicillin. Might have been something as simple as having an ear infection, which in fact could have been a viral ear infection, so the amoxicillin is completely useless. And then subsequent to the amoxicillin, I guess because of the timing or maybe other reasons, maybe the mother was sick as well, the child switched from a breastfed diet to more an adult diet that also had milk. And so you'll notice that above the green arrow, after that timing, you'll see a dramatic drop in the amount of biodiversity in the child's intestines. And so again, anything that's probably just about anything that's happening subsequent to that amoxicillin event is a reestablishment that may or may not be the normal, typical cycle that colonizes the gut in terms of our associations with microorganisms that we co-evolved with. Okay, so people have examined and looked at a variety of things that influence the microbiome. These include whether someone is delivered vaginally or is delivered via cesarean section. And in fact, there are some hospitals that are now piloting and looking at, even if a child is born via cesarean section, to actually swab the mother's vagina and try and transplant those bacteria into the child's mouth. Children that are breastfed tend to have intestinal flora that are more typical and also more diverse than those that are fed by formula. And of course, having early antibiotics changes, at least temporarily, the types of microbiome that is found in young guts as well. And this association here on the chart is one that is one of these interesting epidemiology studies. So on the left, you have, in terms of increasing orange to red color, the obesity trends of adults in the United States by state in the year 2010. And again, this is data coming from the CDC. Again, one thing that's nice about obesity in terms of its study is that you can largely take census data, which includes height, weight, and is generally considered to be reliable data in terms of very large data sets, and you can calculate somebody's body mass index. Now on the right, the map is the density of antibiotic prescriptions. Oh, sorry, yeah, I said BMI, body mass index is what that stands for. On the right, you have the blue map, where the increasing blueness is an increased frequency of prescribing antibiotics. And there is an amazing overlap between the two. Again, it's a correlation, and at this point, not necessarily causation, although there are things that we biologically know in the lab that associate poor intestinal microflora with obesity directly. Again, small scale research and data has shown that there is an association with one's bioenergy related to what type of microorganisms you have in your intestines. That actually can lead to increased risk of obesity. Again, this is a bit of a summary slide, and I'm going to show the data for this next, and that's from a different lab by Dr. Gordon. But the idea here is that you can get this vicious cycle, that if you have an altered gut microbiome, then our body is being invaded by things we haven't co-evolved to play nice with. And if we haven't learned to play nice, then what we do is we create inflammatory signals. And then when you have this low-grade inflammation, this leads to stress signals. And stress signals are things that can also, again, further promote an imbalance of the microbiome. But then additionally, things like cortisol are things that are associated with an overall altered body metabolism as well. And so, again, the problem is that this can be a very vicious forward-acting cycle, that it can basically, you alter the host physiology, creates more stress, alters the microbiome to be more different than it should be, which then promotes more stress, more inflammatory signals. So again, probably one of the foci of future medical interventions would be to try and break that cycle. Okay, so this is a slide that's a bit dense with text. Again, I will be putting this up as a slide share so everyone can see it and read it in their own time. I will summarize the slide. And again, this is from a paper, again, the Jeffrey Gordon Lab, The Effective Diet on the Human Gut Microbiome. And what they did was they took human feces, which, as the paper specifically describes, was fresh, of a healthy adult male. And they froze some of that away. And the rest of it, they both kind of in a raw form, as well as a somewhat, trying to pull out the microorganisms, again, getting rid of all the physical matter, and actually put that into mice that were grown in a very sterile environment. So they had no microbiome of their own. Again, these were mice grown in sterile environment. They were germ-free, and then were colonized with a healthy male's gut. And then the experiment was to feed these mice different diets, either a low lipid, low fats, in other words, as well as high in plant polysaccharides diet, which is what we consider in general a healthy diet, versus a so-called Western diet, which was high in fat, high in sugar. And they did a variety of experiments where they fed mice those different diets and watched what happened to the microbiome, the percentage of different species present, and then also would do things like swap out the diets or actually swap out the fecal matter, basically take fresh mice, do fresh transplants of the fecal matter. And again, the bottom line here on this slide is the summary that I want you to take home from this, is that transplanting fecal matter where, two things. One, the diet can alter the microbiome of the mice, but then also if you take out the microbiome and put it into a fresh mouse, it can actually alter its obesity levels. And so does Simulat have a question? You can go ahead. I'll pause here. Yeah, I find them really interesting too. And again, we actually have this ability through modern techniques to sequence and know large numbers of microbiomes. So again, Simulat asked the question, and again, this is something that I mentioned, but again, aren't mice as infected by microbes as we are? And yes, that is true. But in this laboratory experiment, if you take mice that are freshly born and very carefully raise the mothers in a germ-free environment, and then birth the pups, and then put them in a completely germ-free environment, their stomachs never get colonized. And so you can perform this experiment reliably to recreate a human microbiome in a mouse. And again, the term for that is up here. It's called noozoic, or sorry, nodobiotic. Yeah, I mean, modern research techniques are pretty cool. Yeah, so let me does that answer your question sufficiently? Okay, cool. Yeah, so part of this research was to establish that it was a reliable recreation of the human microbiota in the mice. So again, one thing you can do with really powerful sequencing machines, with computers that sequence and analyze data, is that you can collect lots and lots of bacteria. You don't even have to collect the bacteria. You can basically chew them up, dissolve them in a fluid, and then sequence the DNA to know what you have in there. And so this chart is very dense, and it's a little bit small, so I apologize for that. But again, I was trying to take the original image from the paper. That in the chart on the top right, where you see blue, green, a little bit of orange, and red bars, and the yellow ones, these are indications of which families of bacteria are present in the microbiome and in their relative proportions. Again, they can actually do more. They actually go by the species, but they're grouping them by families. And so what you have on the left is a healthy diet. And you see a lot of blue, a lot of red, a little bit of green, and not a lot of yellow. And so again, what Abby is asking, one introduces fecal matter into the sterile mice. And yes, that is the primary nature of the experiment, is to take human microbiomes derived from a fresh fecal expulsion while the person is also feeling healthy, and then feed them to the mice. And then they actually establish themselves. So again, once they've been put into the mice, they are established and they stay in the mice. They don't go away. And okay, so the third column over, where you see more yellow, more red, and less blue, and a lot more green, that particular column is showing a mouse that originally had a good diet and then was fed a Western diet. And you'll notice that it dramatically changed the overall proportions inside that mouse. And again, the diet has a very strong influence on what sort of microbiome is present in terms of the proportions. And in fact, if you have a Western donor and then feed it a healthy diet, you can revert it back. So again, I'm starting to make you count. 1, 2, 3, 4, 5, 6. The sixth column from the left is a Western diet donor, but then fed a healthy diet. And in fact, it reestablishes to a more healthy looking microbiome. Okay, so the other important thing, and again, I didn't pull the chart from this, is that if you take a Western diet fecal culture and put that into a fresh mouse, then in fact the mouse develops signs and features of obesity, even if it's fed a healthy diet, at least for a little while. And so what this also argues is that these altered microbiota, can actually influence the energy metabolism of the host organism. Okay. And again, so Abby also asked a question about the mice. And again, the key thing is that in terms of experimental design, one is trying to make this a very reductional, easy way to analyze. And again, people have been doing this. And this is a study that was in 2009, I think, and he's followed this up with other work. And again, he's actually interviewed in one of the Science Friday podcasts as well. Okay, so again, let me go to what I think is kind of one of the near future types of ways we're going to approach this. So again, put your eyes close on the screen. And yeah, I kid you not, those are poop pills. Now again, this is a slightly different treatment and design and regimen for a different type of infection. So Clostridium difficile is something that can cause both painful, debilitating, and I think even through dehydration, death. It is a relatively common infection that causes diarrhea that's extremely painful. And again, a lot of people can be treated with antibiotics. They can, again, strive through the disease, just make sure they stay hydrated. But some people have very persistent infections that are very difficult to get rid of. And so the idea, again, based on this catalog of thinking about the microbiome, people have shown for some time that you can do a fecal transplant, which again, yeah, taking poo from one person and putting it into another person, that type of fecal transplant from a healthy donor can help reverse and cure the infection, the ones that are very recalcitrant. And so what is now becoming, again, I got this from a relatively recent paper that's been making the rounds, that perhaps an oral ingestion route is actually the better way to go. And so, again, this is something that's an intractable disease. It's not necessarily a matter of, oh, your energy balance, whether you have asthma or whatnot. But I think here, this is one of these things where at least the literature can medically establish in a nice way that these types of transplants can make not only a difference in what we see, but a very specific difference in the health of patients. And that is by giving the intestines a chance to get colonized by the bacteria that we have co-evolved with, that we have this association where our immune system tries not to get rid of them, or they've learned ways to live happy in our immune system, that this is something that as we expand upon these ideas, we can actually again, alter people's health for the better. Now, as I was making this talk, I really am a fan of keeping things as close within an hour as I can. And so, again, my original design had been to maybe talk about probiotics in the diet and in health. I will say that there are a lot of early indications that healthy diets that specifically include those features that either have live cultures or have the nutrients and the food chemicals that support the happy living, the happy growth of our microbiota, are things that are out there. And there's a lot of anecdotal evidence. I think we are still in the phases of collecting good clinical data. And so I thought at this point, again, we can follow up with the more stringent conversation about probiotics and the relationship between people and humans and talk about some more specific experiments. But I thought I would end it here and just ask people, one, if they have any questions, but also to voice their opinion about what they think about probiotics, about the value of these things both in our diets, as well as these types of interventions of taking vaginal swabs of freshly born children, as well as formula feeding versus breastfeeding and whatnot. So again, I thank you for your time and your attention, and I will